The placenta. Introduction. Only eutherian mammals, like humans, have a placenta. The human placenta is discoid, round and flat in shape. Hemichorial. The chorion is in direct contact with maternal blood. Deciduate. Some maternal tissue is shed during childbirth. It is attached to the uterine wall and connects the mother and fetus through the umbilical cord. The maternal body accepts the fetus without immune rejection. Development. The placenta forms from two sources. 1. Fetal part, develops from chorion frondosum. 2. Maternal part, develops from decidua basilis. Stages of development. 1. 11th day, the blastocyst is fully implanted in the uterus, surrounded by spaces called lacunar spaces. 2. 13th day, stem villi start forming, connecting the chorionic plate to the basal plate. 3. 21st day, a complete blood vessel system forms inside the villi. This connects with the baby's blood circulation via the body stalk. 4. 3rd-4th week, the lacunar spaces merge to form the intervillous space, filled with maternal blood. 5. 6th week, the chorion changes. Chorion frondosum, embryonic side, grows actively. Chorion leva, opposite side, becomes thinner. 6. 12th week, placenta formation is complete. 7. 16th week, placenta continues growing in thickness and size until term. The placenta at term. Gross anatomy, shape and size. Circular, 15 to 20 cm in diameter, 3 cm thick in the center. Weighs about 500 grams, 1/6 of baby's weight. Covers 30% of the uterine wall. Surfaces. 1. Fetal surface, smooth and shiny. Covered by the amnion. Umbilical cord attached at or near the center. Umbilical vessels branch out under the amnion. 80% of placenta is fetal origin. 2. Maternal surface, rough and spongy. Dull red in color, due to maternal blood. Divided into 15 to 20 lobes cotyledons by fissures. Calcified spots may be present, not harmful. Only 20% of the placenta is maternal in origin. Margin. The outer edge of the placenta is formed by fused chorionic and basal plates. Connected to the chorion leva and amnion. Attachment and separation. Attachment. Placenta is usually attached to the upper part of the uterus near the fundus. Anchored by villi connecting the chorionic plate to the basal plate. Separation. The placenta detaches after the baby is born. Separation happens at the decidua spongiosum layer. Summary. The placenta plays a vital role in providing nutrients and oxygen to the fetus. It starts developing by the 6th week and is fully formed by the 12th week. At term, it is a disc-shaped organ that connects to the baby via the umbilical cord. After childbirth, the placenta detaches and is expelled from the body. Structures. Placenta. The placenta has two main plates. Chorionic plate, inner side, facing the fetus. Lined by the amniotic membrane. Umbilical cord is attached here. Basal plate, outer side, facing the mother. Connected to the uterine wall. Has openings for maternal blood vessels. Intervillous space is located between the two plates and contains villi filled with maternal blood. Amniotic membrane. A thin, single layer of cuboidal cells. Loosely attached to the chorionic plate. Does not take part in placenta formation. Chorionic plate. Composed of 1. Primitive mesenchymal tissue, contains umbilical blood vessels. 2. Cytotrophoblast layer. 3. Synchytotrophoblast layer. Stem villi arise from this plate. Basal plate. Composed of 1. Decidua basilis, maternal tissue. 2. Niedibuch's layer, prevents excessive placenta invasion. 3. Cytotrophoblastic shell. 4. Synchytotrophoblast. Contains spiral arteries that bring maternal blood into the placenta. Forms septa partitions dividing the placenta into cotyledons, 15 to 20 lobes. Intervillous space. Area between the chorionic and basal plates. Filled with slow-moving maternal blood. Contains branching villi, which absorb nutrients and oxygen for the fetus. Stem villi. Extend from the chorionic plate to the basal plate. Develop in 3 stages. 1. Primary villi. 2. Secondary villi. 3. Tertiary villi, functional unit of the placenta. Each cotyledon contains 3 to 4 major stem villi. Surface area for exchange, 10-14 meters squared. Fetal capillaries inside villi are about 50 kilometers long. Structure of a terminal villus. Early placenta. 1. Synchytotrophoblast, outer layer. 2. Cytotrophoblast. 3. Basement membrane. 4. Stroma, connective tissue plus fetal capillaries. Term placenta. Thinner synchytotrophoblast allows easy exchange. Thicker basement membrane. Hofbauer cells, phagocytes that trap maternal antibodies. Placental circulation. Placental circulation consists of two separate blood flows. 1. Uteroplacental circulation, maternal blood. 2. Fetoplacental circulation, fetal blood. Uteroplacental circulation, maternal circulation. Maternal blood enters the placenta via spiral arteries, 120 to 200 in number. Intervillous space holds 150 milliliters of maternal blood at a time. Blood is completely replaced 3 to 4 times per minute. Pressure in intervillous space, 10 to 15 mmHg, relaxed uterus. 30 to 50 mmHg, contracting uterus. Venous drainage occurs through uterine veins. Trophoblast invasion. Extravillous trophoblast (EVT) helps remodel spiral arteries for better blood flow. EVT has two types. Endovascular EVT invades inside arteries and replaces lining. Interstitial EVT invades into the uterine muscle. Defective invasion can lead to pregnancy complications (PIH, IUGR). Fetoplacental circulation, fetal blood flow. Two umbilical arteries carry deoxygenated blood from the fetus to the placenta. Blood enters chorionic plate, moves into the villi, and exchanges oxygen, nutrients. Oxygenated blood returns to the fetus via the umbilical vein. Fetal blood flow through placenta, approximately 400 milliliters per minute. Countercurrent exchange. Maternal and fetal blood move in opposite directions, improving exchange. Placental barrier, placental membrane. Separates fetal and maternal blood but allows exchange. Early pregnancy barrier, 5 layers. 1. Syncotiotropoblast. 2. Cytotropoblast. 3. Basement membrane. 4. Stroma. 5. Fetal capillary endothelium. Near term, it becomes thinner for better exchange. Specialized areas in terminal villi. Alpha zones, thin areas, right-pointing arrow gas exchange. Beta zones, thick areas, right-pointing arrow hormone production. Thickened barrier seen in IUGR, intrauterine growth restriction. Smoking mothers.